Hello everybody! Today, we'll be presenting a study on scalable xeno-free expansion of mesenchymal stem cells in a fixed-bed bioreactor, in collaboration with Bill King from Rooster Bio and Thomas Robert from Univercells Technologies. Now, let's start with the details of the case study.
Hello, my name is Bill King, and I'm Director of Business Development for Rooster Bio. For those that don't know us, or even those that do, I'd like to explain who Rooster Bio is and what we do. As a broad overview, we were founded in 2013 with the mission to fuel the rapid implementation of scalable advanced therapies. We currently have somewhere north of 50 employees and are located in Frederick, Maryland, which is part of the Mid-Atlantic Biotech Corridor. At Rooster Bio, we have lab space for research and development, manufacturing space for products, and warehousing for storage and shipping. A core part of our offerings are cGMP media and cells supported by FDA master files. These are currently being used as ancillary and/or raw materials for multiple ongoing and planned clinical trials. The industrialization of the MSC supply chain has played a crucial role in advancing the field. Standardized cell bank product formats and scalable manufacturing processes have made MSCs more accessible and cost-effective for researchers and developers. Numerous MSC products have received market approval globally, demonstrating their therapeutic value and commercial viability. A new generation of MSC therapies, which we refer to as MSC 2.0 applications, involve gene-modified cells, extracellular vesicles, and other exciting applications of MSCs. These advancements, along with further industrialization of the supply chain, are expected to drive innovation and reduce costs, ushering in a new era for MSC advanced therapies. In order for us to accelerate the implementation of scalable advanced therapies, we need to transform MSC and exosome production by optimizing raw materials and processes to meet your future needs, and so you can incorporate those plans into what you do today. Rooster Bio's products, processes, and development and analytical services combine to accelerate your project process development in several ways. First, we make MSCs and exosomes easy to establish within new programs by providing well-defined raw materials for research and development, supported by easily implementable protocols to get up and running quickly. These products and processes are built to scale, enabling a seamless transition to bioreactor platforms. Utilizing our years of bioprocessing expertise, we function as a process development lab for many therapeutics companies, but we can also provide custom-built MSCs, EVs, and analytical services tailored to your programming needs. These things, combined, allow us to do the heavy product and process lifting while you use your intellectual capital to solve difficult problems. Now, I'd like to hand the presentation over to Dr. Thomas Robert, Senior Innovation and Product Manager at Univercells Technologies, for background on Univercells Technologies and a review of our collaboration.
All right, thank you, Bill. So on my side, before deep diving into the case study, I would like to present our company and our technology. And so Univercells Technologies is a company that delivers bioprocess technologies to a wide range of users for different applications, but mostly in the field of vaccines and advanced therapies manufacturing. We have broad application across the ATMP landscape. We work with a wide range of organizations, such as CDMOs and service providers, but also a large amount of developers, as well as some public institutions. And we have seen that our technology has been used all the way from R&D to phase 3 clinical trials. Actually, we've soon a commercially approved product for a different range of applications, mostly vaccine and gene therapy manufacturing, such as AAV and lentivirus, but more recently, also increasingly with mesenchymal stem cells and other types of stem cells, either for production of these cells as a product for cell therapy applications or for the production of extracellular vesicles or exosomes. And so we are quite happy to see this growing range of applications with our technology. Now on this slide, you can see the ScaleX bioreactor range. And as you can see, it is a relatively wide portfolio of fixed bed bioreactors that go from the Scalex Nexo with 0.5 square meter of surface area all the way to 600 square meter of surface area with the Scalex Nitro bioreactor. And this is a really interesting range of bioreactors with different types of systems that enable users to go from the proof of concept and initial R&D and process development all the way to clinical and GMP manufacturing with the largest scale systems. And we really see this range as a scalable option for low-cost cell and cell-based manufacturing. And now deep diving a bit more into the design of the bioreactor itself. So the Scalex bioreactor is in essence a structured fixed bed bioreactor that enables to grow the cells within a matrix that entraps the cells and enables them to grow at a very high density. And what you can see here on the drawings is that these cells are entrapped within a very homogeneous matrix, and they will be gently perfused in a continuous way with media, enabling them to grow at very high density. But what the Scalex technology is interesting about is that it enables to do this at a very reduced working volume. And so this means that the cells are entrapped in very small working volumes, which gives a lot of flexibility in terms of process configurations. But what this also means is that it enables us to have an excellent linear scalability from the smallest bioreactor design to the largest. And this will really make it much easier for an organization to define process conditions at small scale and actually translate those across the scales to accelerate the process development and scale up. And of course, because these are bioreactors, they offer automated operation and process control, but they also do this at very low footprint. And so thanks to the very low footprint and linear scalability, this really enables us to strongly reduce the cost of goods per dose, which in the end is one of the most important criteria for these advanced therapies, but also shorten the time to market, which is an essential criteria, considering how we want to make these advanced therapies more affordable and more accessible to patients. We really see these two criteria as the most important ones. All right, so now deep diving into the case study itself that we performed with Rooster Bio. So in this case study, what we did is we simply took Rooster Bio's best-in-class xeno-free MSC platform, and we attempted to transfer it into the Scalex Hydro bioreactor, which is one of our R&D bioreactors with a 2.4 square meter surface area. And so we took Rooster Bio's xeno-free human bone marrow stem cells. We performed a seed train in flasks before going into the Scalex Hydro bioreactor, in which we expanded the cells using the Rooster Nourish xeno-free expansion media for 6 days with a day-free bolus of Rooster Replenish xeno-free media. And after this expansion period, we tried to collect the cells from the bioreactor using the Scalex Cell Collect module. And this Cell Collect module is, in a sense, a vibration platform that enables to induce a mechanical activity on the bioreactor to support enzymatic detachment from the fixed bed. This enzymatic detachment itself is a relatively standard protocol. We performed between 50 to 45 minutes incubation using EEE at 37 degrees Celsius. And then we harvested using the Cell Collect modules. We also tried different vibration frequencies to see how that could impact the efficiency of harvest. And of course, at the end, what was really important for us was to do an extensive quality control to check not just the count and viability of the cells, but also the quality of the cells, either in replating assays or in a FACS characterization. And we also did some confocal microscopy to actually have a look at how the cells grew inside the bioreactor. All right, so on this slide, you can see how the cells were able to attach rapidly inside the Scalex bioreactor after inoculation. On the left, you have a table that shows all the data that we have for the 3 runs that we performed. And in a sense, what you can see is that after 60 minutes of inoculation, all the cells have attached on the fixed bed, meaning they are no longer detectable in the supernatant inside the bioreactor, which is also why we cannot measure viability at that stage. But what we also saw is that the viability in the initial stage of this inoculation was very high. So overall, very strong data suggesting that the cells can be very fastly inoculated inside the fixed bed bioreactor. And on the right, you can see with the graph an illustration of these kinetics, showing how basically after 15 minutes, most of the cells are already attached. And then by the end of the hour, we cannot detect them anymore in the bioreactor. After that, we looked at the cell growth for up to 6 days inside the bioreactor. And there, one of the easiest way to monitor the cell growth inside fixed bed bioreactor is to look at metabolites, such as glucose and lactate, and simply measuring the concentration of these metabolites inside the bioreactor to extrapolate metabolic activity and therefore cell growth of the cells inside the bioreactor. And what you can see here is that for the 3 runs, we had a typical steady decline in glucose over the course of the expansion, which was correlated with an increase in lactate concentrations, as you can see here. One of the things that you might also see is that for the third run that we did, we tried a different strategy in terms of glucose concentration. We started off with a lower glucose concentration and then perform a bolus at day 5 to ensure sufficient feed of the cells. And so this is, which is why you see that the curve is a bit different here. But of course, if we look at the data in a different way, for example, look at glucose consumption and lactate production, we had quite comparable profile across the 3 runs for either glucose consumption or lactate production. Another way to look at the cell growth is actually to take samples of the fixed bed itself during the expansion, and this can be done with the ScaleX Hydro bioreactor, which is an R&D system. And so we could actually take samples to perform cell counts during the expansion. We did this mostly over the last 3 days of the expansion to monitor the growth of the cells and their ability to attach on the fibers. And so this is always interesting to do. And of course, we took the opportunity to actually do some confocal microscopy on those fixed bed samples to really see how the cells could attach to the fixed bed fibers. And we were really surprised and happy to see that the cells were showing a very nice morphology of sticking to the fixed bed fibers and actually making a very homogeneous monolayer. Even though it's in 3 dimensions inside the fixed bed, we could really see that they were organized in a very nice monolayer across the fixed bed samples that we took. Now, of course, taking fixed bed samples to count the cells is always interesting to do, but one would want to try to avoid this, I would say, in a routine manner to save time and costs, or when moving to more clinical applications to avoid performing open manipulations of the bioreactor. And so for this, relying exclusively on glucose and lactate is the way that we recommend to do. But to make this easier, we have developed a tool which is called Scalia Vision, and it's in a sense a software tool that we can offer to any customers and user of ScaleX bioreactors. And this software is very interesting because in a sense it enables to estimate the biomass or the cell number inside the bioreactor based on glucose and lactate measurements. But it's not just a tool to estimate the cell number at a given time, it's also a predictive model, very powerful one, that enables you to estimate when you will reach, let's say, a specific cell density target. If you want to target to have a specific amount of cells inside the bioreactor, this tool enables you to understand when is the best time to harvest, for example. And so we were quite happy to pilot the use of this Scalia Vision with mesenchymal stem cell expansion during this case study, and showing that we could really perform predictions using the Scalia Vision model and have very good correlation and very good accuracy with the data that we could actually get from the cell sample counts. And then, of course, at the end of the expansion phase, we performed the harvest procedures for each of the 3 runs that we did in this case study. And what you could see is that between run 1 and run 2, we were really able to nicely increase the yield by making some adjustments, mostly increasing enzyme incubation and vibration frequency. And then run 3, we did some more tweaks, and we were able to achieve also comparable results, if a bit lower with run 2. But overall, with these N=3 runs, which is of course a limited number of runs that we perform, but we can really show that it's possible to increase and optimize harvest yields from the fixed bed bioreactor. We do believe that we have further room for improvement in these applications, and it's likely that it will be very much process dependent, depending on the cell line and the process conditions used. But overall, we see this as a very strong proof of concept that mesenchymal stem cells can be grown and harvested efficiently from the ScaleX bioreactor. And of course, most importantly, the viability of the cells that we harvested from the bioreactor was really excellent, consistently above 96%. So this is, of course, very important for us to see. And just to illustrate what this means in terms of scalability, As you can see, we were able to harvest, in this case, up to 460 million cells. And so that means that with a single vessel, a small hydro vessel of 2.4 square meter, we actually could duplicate the equivalent of growing cells with more than 46 T175 flasks. So this is just a simple illustration of the extent of scalability that we are able to achieve with ScaleX technology, knowing that there's further room for improvement on this, and that there are other models of bioreactor that we offer, for example, up to 30 square meters with even further scalability possibilities. And then finally, and maybe most importantly, what we really wanted to ensure is that the cells that we would harvest from the bioreactor were actually relevant cells in terms of phenotype and potentially in terms of therapeutic activity. So what we did is we performed a comprehensive FACS characterization assay to look at the markers of these cells in comparison with the T-flask controls, and we did this for each of the 3 runs that we performed. And what you can see is that on the top 2 graphs, either from freshly harvested cells or from replated cells, we had consistently very high expression levels of all the markers that one would expect to be present in mesenchymal stem cells, at least always comparable to flask control, while the markers that one would expect to be negative were always as well on the low side. And on the other thing that we wanted to see with this quality of the cells is the impact of vibration frequency. And so on the lower graph, you can see that we tested in this case for the first run, we tested 50, 60, and 70 hertz of vibration just to make sure that the cells were not harmed by this extent of vibration during the harvest. And as you can see, this is not the case. The vibration does not have a detrimental effect on the phenotype of the cells, similar to what we saw with the viability, for example. So this brings me to the conclusion of this slide deck and of this webinar, and I hope that we could convince you through this data that ScaleX bioreactor can be used as an efficient cell expansion platform for mesenchymal stem cells and other types of stem cells. And in this case study, we really leverage Rooster Bio's expertise and best-in-class platform with their xeno-free stem cells, media, and regions to perform a comprehensive process transfer very simply into the ScaleX hydro bioreactor, and tested the ability to expand the cells, harvest them, while looking at the quality attributes. And what we could really see is that this process transfer was successful. It's a very strong proof of concept that the cells can be efficiently harvested from the bioreactor after a successful expansion, while meeting the key quality attributes, which of course is most important. And so we really believe that thanks to its scalable nature and cost-effective nature, ScaleX bioreactor can really be very attractive options for expansion of stem cells for therapeutic purposes. So with that, I would like to thank you for your attention. And if you would like to know more about our technology, of course, feel free to reach out to Univercells Technologies or to Rooster Bio for their products and services. Thank you very much.